Hi, Physiology class. Allow me to introduce Zach Nephron. Today we're going to talk about the loop of Henle and its role in helping to conserve water. So the filtrate at this point, having gone through filtration and tubular reabsorption, is still not very concentrated. It contains a large amount of water, and we can't afford to pee out all of that water. So we need a process to get it back. And we're going to use salt to get that water back. So the best way to move water around the body is to move salt first and let the water follow that salt by osmosis. So we're going to create a salt gradient inside the loop of Henle, and we do that by pumping the sodium into the medulla of the kidney. And then as the medulla becomes saltier and saltier, it's going to draw that water towards it by osmosis. The water will move from a high concentration into the low concentration of water area. So we're going to move water out of the descending limb of the loop of Henle and the far end of the distal convoluted tubule, and that'll get it, once it's into the medulla, that'll get it back into the blood so it's not secreted. So this is the loop of Henle. This is the descending limb here and the ascending limb over here. And the process that's occurring in the loop is called the countercurrent multiplication mechanism. So what we've got inside of our loop here is the concentration of our filtrate. So the 300 milliosmoles is less concentrated than 600 right here. The 900 is even more concentrated. So at the very bottom of our loop, it's very concentrated, 1200 milliosmoles. And then as we go back up this side over here, we can see it's getting less concentrated again. So that's the countercurrent part that they're referring to in the name, is that this concentration is taking this hairpin turn and we're changing our molarity around that turn there. So the descending limb, the downward part of the limb, is permeable to water. But the ascending limb, the point going up here, is impermeable to water. So what we're doing to move the water is we're running sodium and chloride pumps on the side of the ascending limb. That'll pump salt into the medulla, the area that's around this loop here. So the medulla is getting saltier and saltier and saltier all over the place out here. And then that's going to draw the water from where it can leave, which is out this descending limb side. The water will leave, whoops, maybe make the water, would have been logical to make it blue, but I chose red. So the water is now leaving the descending limb side because it can get out over there. And so I'm pushing the water back into the medulla, and then the blood, the paratubular capillaries that are all around that loop will start to pick up that water and send it back into the circulation. So here's the loop of Henle, and the countercurrent mechanism is at work there, drawing water back into the medulla, so that's how we can concentrate our filtrate. If we're dehydrated, we can, under the control of a hormone, which we'll talk about later, our diuretic hormone, we can make this part of the tubule, the collecting duct part of the tubule, also permeable to water. And that gives us a second chance to capture that water for those times when we are dehydrated and we really, really need to concentrate the urine.
As the filtrate moves down the loop of Henle, the concentration of solutes increases in the interstitial fluid surrounding the nephron tubule. Since this portion of the tubule is permeable to water, water leaves by osmosis and is reabsorbed into the blood. This concentrates the filtrate. In the ascending loop, the surrounding fluid becomes more dilute. This portion of the loop is impermeable to water, but not sodium chloride. Sodium chloride diffuses out, lowering the solute concentration of the filtrate and adding to the solute concentration of the surrounding fluid. Near the top of the loop, sodium chloride is actively transported out, further diluting the filtrate. More substances are reabsorbed from the distal tubule. Sodium chloride is actively transported out of the filtrate. Bicarbonate ions may be reabsorbed too, helping to regulate body pH. Some drugs and poisons are secreted from the blood into the filtrate.
The last step in the filtrate formation that we want to talk about is tubular secretion. And this does happen along the whole length of the tubule, but it's happening the most in the distal convoluted tubule. So that's what we're going to focus our attention on. So this gives us a second chance to get rid of the bad stuff. So if we didn't end up catching some of the waste products in the beginning of the filtration process, now is our chance to dump them from the blood into the renal tubule so that we can pee them out. So substances are secreted in the distal convoluted tubules from the blood, and some of the secretions or waste in the greatest number, some of those things are urea, which if you recall comes from the breakdown of protein metabolism, urobilin, which is a pigment we get from recycling red blood cells, potassium ion, hydrogen ion, because remember we're always becoming acidic and we need to get rid of hydrogen ion. So a number of things we put in our bodies, like medicines that we take, like antibiotics, penicillin, ibuprofen, so drugs that we need to get rid of, medicine type drugs plus recreational drugs, marijuana, cocaine. That's why they do drug tests and check your urine, right? Because we can find those drugs in your urine as your body's trying to get rid of the toxin. This is a good strategy for helping us to maintain our pH. Remember, we need to be at 7.4. We talked about how the respiratory system can help us do that, help us regulate with bicarbonate ion. And here is a chance to help us get rid of the hydrogen directly by peeing it out. So for most people, urine is acidic because mostly our bodies become acidic as the day goes on. So this graphic has a side-by-side look at the process of tubular reabsorption and compares it to tubular secretion. So here we have tubular reabsorption, and in this process we're taking from the filtrate the good things that we still want. We want to use them in our body, but we have to catch them and put them back into the blood. So here we're moving from the tubule and putting back into the bloodstream. So we're reabsorbing it into the blood. In the process over here on the right, this is tubular secretion. So what we're doing here is we're taking, here's tubular secretion, we're taking from the blood the stuff that's bad, the waste, the toxins, and we're sending them back into the bloodstream. So we're trying to get rid of them. So in the distal convoluted tubule, here we are, distal convoluted tubule, we have to be careful about our ion movement, the number of positives, negatives that we're moving here. The body tries to keep that ion charge balanced. So if in this picture example here, we're moving sodium into the blood, it's a positive ion. What we're also going to have to do is move potassium back out of the blood, or hydrogen ion because it's also positively charged. So the potassium or hydrogen would end up going into the urine so that we can keep that charge balanced the same. So a rule of thumb that you should be paying attention to is that sodium and potassium usually go opposite directions of each other, but water will always follow the sodium. So let's review real quickly the three different processes that we talked about in forming the urine filtrate. So the first step was glomerular filtration, where in the glomerulus we're separating the small components of the blood from the bigger components. The big substances like the blood cells, the large proteins like hormones and enzymes, they're going to stay in the blood. They're too big to fit into the glomerular capsule, sieve. And the smaller substances, small nutrients, small molecules like water, sugar are going to go ahead and move into the filtrate. So that was glomerular filtration, just separating small from big. And then we went into tubular reabsorption. And in tubular reabsorption, the kidney is trying to catch a lot of those good things, nutrients that just now got into the filtrate, and try to catch them and put them back into the blood because we don't want to lose them. So those would be those small nutrient molecules like glucose and amino acids, important ions like calcium and potassium and sodium. So we want to get those substances back. So we reabsorb them back into the bloodstream. And then we just talked about tubular secretion, which was another chance to remove the toxins and the waste from the blood and send it into the renal tubule or the nephron so that we can pee it out. And so those would be waste products from metabolism like urea and urobilin, plus drugs, excess ions. So in secretion, we get a second chance to remove the waste.
Nephron, structure and functions. Objective: to understand the structure of a nephron in detail. The excretory system in human beings consists of a pair of kidneys. A magnified view of the longitudinal section of kidney shows two prominent regions, a cortex and a medulla. As we zoom into the cortex region, it shows the presence of numerous nephrons. Each nephron has a Bowman's capsule, a proximal convoluted tubule, a loop of Henle, a distal convoluted tubule that leads to a collecting duct. Nephrons are convoluted in the cortex region and straighten out as loops in the medulla region. Each nephron has a Bowman's capsule, which is a cup-shaped structure that has a layer made of squamous epithelial cells. In the cavity of the Bowman's capsule, there is a bundle of capillaries known as the glomerulus. The blood vessel entering the Bowman's capsule is known as the efferent arteriole and is thicker than the blood vessel exiting the capsule, which is thinner and is known as the efferent arteriole. The Bowman's capsule and the glomerulus together form the Malpighian body. This capsule extends into the proximal convoluted tubule, PCT, on the inner side. This tubule is lined by a brush-bordered epithelium with numerous microvilli. This PCT leads into the loop of Henle, which lies in the medulla region. It is U-shaped and has a descending limb and an ascending limb. This further leads into the distal convoluted tubule, which is again seen in the cortex region, and it finally joins the collecting duct. As blood flows through the glomerulus, water, salts, and waste diffuse into the Bowman's capsule. This liquid then passes into the PCT and the loop of Henle, where water and important salts are absorbed back into the blood vessel. Only the waste material like urea and some water molecules proceed into the DCT, where more water may get absorbed into the interstitial fluid. This liquid formed is the urine, which is released into the collecting duct. This finally trickles through the kidney into the ureter, which helps the passage of urine into the urinary bladder. After a while, the urine can be excreted from the bladder. To summarize, the excretory system in human beings consists of a pair of kidneys. The longitudinal section of kidney shows two prominent regions, a cortex and a medulla. Inside the kidneys, we observed numerous nephrons. Each nephron has a Bowman's capsule, a proximal convoluted tubule, a loop of Henle, a distal convoluted tubule that leads to a collecting duct. In the cavity of the Bowman's capsule, there is a bundle of capillaries known as the glomerulus. As blood flows through the glomerulus, water, salts, and waste diffuse into the Bowman's capsule. This liquid then passes into the PCT and the loop of Henle, where water and important salts are absorbed back into the blood vessel. Only the waste material like urea and some water molecules proceed into the DCT, where more water may get absorbed into the interstitial fluid. The liquid remaining in the DCT is the urine, which is finally released into the collecting duct.
Okay, so let's take a look at this table here. So we have on the table the plasma, which is the blood, the glomerular filtrate, which is our filtrate at the beginning of the urine formation process, and then here's our final product, the urine. So let's take a look at some of these substances and see what's happening to them on their way through the nephron. So sodium starts off with 142 milliequivalents per liter in the plasma. All of that ends up in our filtrate, 142, but in the urine we have 128. So some of that sodium was pulled back into the blood. We're not peeing it out. So that process would be tubular reabsorption of the sodium. So we took some of that sodium back. And if we look at, let's look at phosphate down here at the bottom. It starts off at 2 milliequivalents per liter. So in the blood there was 2. In the filtrate there's 2. But when we look way over here at our product, it's now 40 milliequivalents per liter. So that means I must have done a lot of tubular secretion with the phosphate because a whole bunch of it ended up in my urine, but I didn't initially capture 40 at this point in my filtration process. So I had to have secreted it later on in the distal convoluted tubule. Glucose down here. Let's look at the glucose. So glucose, we had 100 in the plasma, got all of it in the filtrate, 100 in the filtrate, 0 in the urine. So that tells me I'm doing what to that urine? Tubular reabsorption, tubular reabsorption. Creatinine was 1 in the plasma, 1 in the filtrate, and then 196 in the urine. So that tells me creatinine is a waste product. My body's trying to get rid of it, and I am actively secreting it along that nephron tubule to try to get it out of my body. Here's a comparison table from your book to go through the 3 processes. So water, we filter 180 liters of water per day, and then we reabsorb 178 of those liters. So all we're losing through excretion or urination is about 1.8 liters of water. So we're recapturing nearly all of it. Only 1% is left. 99% is reabsorbed. The sodium, we started with 630 that we're filtering. We reabsorb a lot of it, and we're excreting only about 0.5%. That's average for people in general. Americans have a very salty diet, so we actually excrete more than that 3.2 grams. Thanks guys for listening. I will be back tomorrow and be there to answer any of your questions. I know this can be a little confusing. There are some more videos on Blackboard, Khan Academy, and some other video lectures if you still need some help in these processes. Have a good day.